science fans, and welcome to SHE-ensya. Our topic today is the story behind DNA sequences. It is the decade of biotechnology, and we are seeing developments at an amazing rate. Nucleotide extraction and processing has become standard lessons taught in biology courses worldwide. And there are vast applications embedded within these sequences. But after the precise and tedious process of DNA extraction, what can we then learn from the biomolecule that we now have? One of the ways by which information from the DNA can make sense is through the process of DNA fingerprinting. There are short tandem repeats, or STRs. These are repeating sequences within the genome that are 2 to 7 base pairs in length, with the number of repeats varying among individuals, making STRs effective for human identification purposes. DNA from tissues as little as 1 nanogram is extracted, and then the corresponding STRs are amplified. The amplified STRs can then be visualized and analyzed using agarose gel electrophoresis or fluorescent marker technologies. In order to utilize STR information as a means of human identification, law enforcement agencies worldwide have established the frequency with which each allele or version of each of the 13 core STRs naturally occur in people of different ethnic backgrounds. The use of STRs as a means to identify a person based simply on their DNA is dependent on how we inherit half of our genes from each of our parents. For instance, the STR known as D7S820 found on chromosome 7 contains between 5 and 16 repeats of GATA. Therefore, there are 12 different versions possible for the D7S820 STR. A person with a D7S820 gene with 10 repeats and 15 repeats would have inherited the version with 10 repeats from one parent and the version with 15 repeats from the other parent. And because there are 12 different versions for this STR alone, there are therefore 78 different possible combinations. And this is just for one of the 13 standard STRs used for human identification. Another way to find meaning out of DNA sequences is to use the unique mutations found within genes that can be recognized by restriction enzymes. A restriction enzyme is an enzyme produced chiefly by certain bacteria and having the property of cleaving DNA molecules at or near a specific sequence of nucleotide bases. There are 4 general types of restriction enzymes. They differ based on where they cut the DNA based on a sequence of nucleotides called recognition sequences and the type of sequences they can read. And based on the versions of the genes that we inherit, these can be cut up by restriction enzymes and result to different sized fragments. The variable locations of these sites that can be recognized by restriction enzymes and the resulting different sizes of DNA fragments can be visualized using agarose gel electrophoresis and be used to detect the identity of a person from tissue left behind. This process is known as restriction fragment length polymorphism, or RFLIP. It can be used to detect paternity, identify the species of samples collected from the field, or even detect genetic variability of individuals within a population. The use of STRs and RFLIP take advantage of our knowledge of DNA sequences, but primarily they use sizes of DNA fragments as a means to generate conclusions from data. But we can also determine the actual sequence of nucleotides from our samples and use it to create our own different analysis. The conventional method of determining the sequence of nucleotides in a DNA extract or PCR product is called Sanger sequencing. The DNA sequence of interest is used as a template for a special type of PCR called chain termination PCR. Dideoxyribonucleotides lack the 3' OH group required for phosphodiester bond formation, and they are also fluorescently labeled. When they attach, extension stops. The result of chain termination PCR is millions to billions of oligonucleotide copies of the DNA sequence of interest terminated at random lengths by the dideoxyribonucleotides. The visualization of the different sized fragments can be done using agarose gel electrophoresis. With agarose gel electrophoresis, the nucleotides will be arranged from smallest to largest, reading the gel from bottom to top. The last step simply involves reading the gel to determine the sequence of the input DNA. Because DNA polymerase only synthesizes DNA in the 5' to 3' direction, each terminal ddNTP will correspond to a specific nucleotide in the original sequence. Therefore, by reading the gel bands from smallest to largest, we can determine the 5' to 3' sequence of the original DNA strand. The data output of DNA sequencing is called a FASTA file. The FASTA format is a text-based format for representing either nucleotide sequences or peptide sequences, in which base pairs or amino acids are represented using single-letter codes. A sequence in FASTA format begins with a single-line description followed by lines of sequence data. So what can we do with FASTA files? The simplest but vital process is to do a BLAST search. Doing a nucleotide BLAST search compares the nucleotide sequence that you have with the millions of sequences in reference at the National Center for Biotechnology Information Database. It uses algorithms to check for uniqueness of the sequence and the likelihood that the sequences match up by coincidence. At its simplest, a BLAST search will give you a list of species or organisms to which the DNA sequence is most likely to belong to. The results will contain parameters indicating how many of the nucleotides match those in the database, the probability that the match was caused by a coincidence, and the confidence of the algorithm that it created the correct match. BLAST-N can also show you which portions of the nucleotide sequence match the reference through the alignment scores visualization. Doing a BLAST search is crucial because it confirms if you were able to amplify the correct sequence. Gene sequences are also useful for detecting evolutionary relationships between organisms, and this falls under the discipline of phylogenetics. Phylogenetics in biology is the study of the ancestral relatedness of groups of organisms, whether alive or extinct. Parts of a phylogenetic tree include the tips of the tree branches representing the taxa in the study, and the taxa may be at any taxonomic level including orders, species, or populations. The lines within the tree are called the branches, and the points at which branches connect, or the tips of the branches, are both called nodes. Internal nodes connect branches, while external nodes are the tips that represent the taxa. Some trees will have a basal node known as the root, while a grouping of an ancestor and all of its descendants is known as a clade. So how do you generate a phylogenetic tree from gene sequences? There are a variety of programs that you can use, but they all share a fundamental process. Evolutionarily related species have a common ancestor. Before they split into separate species, they had exactly the same DNA. But as species evolve and they diverge, they will accumulate changes in the DNA sequences. And we can use these differences in the genetic sequence to approximate how closely related two organisms are. The less differences there are in the gene sequence, the higher the likelihood that they are closely related. But the more differences there are, the more likely that they are distant relatives. The first thing done is the alignment of the DNA sequences that you're going to compare. And make sure that you're comparing the same gene, otherwise it's comparing apples to oranges. The program's algorithm then compares the sequences to see how similar they are to each other. For example, Sequence 1 and 2 may have one difference, while sequence 2 and 3 have two. Sequences 3, 4, and 5 may have 5 differences, and so on. The program then calculates how similar any two sequences are and finds the sequences with least differences and groups them together. It then separates those with the most differences. The program then draws a phylogenetic tree that represents those evolutionary relationships. The nodes in the tree represent how close or far the genetic relationships are between the organisms, whereas the length of the branches represents when those organisms diverged from each other. In phylogenetics, your results will vary dependent on the nucleotide region that you're using. The more regions you use, the more accurate you tend to be. But no single region will give you the full picture. And due to this need for other informative regions in the genome, there are other markers that can be used. Repeating units called single nucleotide polymorphisms and microsatellites found across larger portions of the genome can be used to detect identity and evolutionary relationships with higher accuracy and precision. Due to the variety and the large number of these regions that are needed to be amplified, the use of traditional Sanger sequencing would be too time-consuming and costly to be useful. Lucky for us, there is now a new way of sequencing DNA, which is called next-generation sequencing. Next-generation sequencing is significantly cheaper, quicker, more accurate, and reliable than Sanger sequencing. It does this in two ways. Firstly, in NGS, the chemical reaction may be combined with the signal detection, whereas in Sanger sequencing, these are two separate processes. Secondly, and more significantly, only one read at a maximum of 1 kilobyte can be taken at a time in Sanger sequencing. But in NGS, it is massively parallel, allowing 300 gigabytes of DNA to be read on a single run on a single chip. The output of next-generation sequencing is not a neatly arranged sequence of nucleotides that are already in the correct order. It is a set of possibly overlapping fragments that you now have to process using bioinformatics in order for them to make sense. The process is almost like solving a jigsaw puzzle. Except that for this jigsaw puzzle, occasionally you won't have a guide picture, and most of the times, not all pieces of the puzzle are available. Even now, newer sequencing technologies are being developed, and that's why next-generation sequencing is really more of now-generation sequencing. And soon enough, we will have faster and cheaper methods in order to make sense of the story behind the DNA. I hope you were able to learn something in this short video about the stories behind DNA sequences. I hope you can give this a thumbs up and also subscribe to our channel. If you have any comments, questions, or suggestions, please don't hesitate to message me, your resident Filipino scientist, in the comments section below. Thank you very much and see you around!